For over 40 years, the world's most brilliant minds have run into the exact same brick wall. We have developed incredible drugs that can reduce HIV to undetectable levels, allowing people to live long, healthy lives. But a true, permanent cure? It has remained completely out of reach. Why? Because HIV is a master of biological hide-and-seek. Unlike other viruses that simply float around your bloodstream, HIV slips inside your immune system's T cells and literally stitches its own genetic code directly into your DNA. Then, it goes completely silent. It creates what scientists call a latent reservoir. Antiretroviral drugs can kill the active viruses in your blood, but they are totally blind to these sleeping cells. The moment a patient stops taking medication, the hidden virus wakes up, multiplies, and mounts a fresh attack. To destroy it, we needed a miracle. But instead, we got a team-up of two of the most revolutionary technologies of the 21st century. Front number one, nanotechnology. If the virus is a smart predator, we had to build a better trap. Scientists are now engineering ultra-microscopic particles known as nanotraps. These aren't living cells, they are synthetic, biodegradable nanoparticles coated with the exact same cellular receptors that HIV looks for when it wants to infect a human T cell. To the virus, these nanotraps look exactly like its favorite target. It's a microscopic Trojan horse. The virus locks onto the nanotrap, thinking it's about to hijack a real cell, but instead it gets permanently stuck. The nanotrap acts like biological flypaper, completely neutralizing the free-floating virus and preventing it from ever infecting your real immune system. Your body then safely clears away the trapped virus particles naturally. By deploying these decoys, we can aggressively neutralize circulating viruses without harming a single human cell. But what about the viruses already hiding inside the DNA reservoirs? That brings us to front number two, CRISPR gene editing. Specifically, a historic clinical trial codenamed EBT-101. While nanotraps handle the active invaders on the outside, CRISPR is sent inside to perform a genetic exorcism. CRISPR acts as a biological GPS paired with molecular scissors. Scientists program it with the exact genetic signature of the hidden HIV virus. CRISPR travels through the body, scans the DNA of infected reservoir cells, locates the sleeping viral code, and cleanly snips it out. Once the viral DNA is excised, the cell's natural machinery repairs the strand, leaving behind a perfectly healthy, virus-free human cell. The EBT-101 trials are currently testing this exact mechanism in human patients to answer the ultimate question: can we safely wipe out the latent reservoir once and for all? When you look at these technologies individually, they are remarkable, but together, they create a perfect pincer movement. Nanotechnology clears the battlefield on the outside, protecting healthy immune cells from new infections and mopping up active outbreaks. Meanwhile, CRISPR infiltrates the enemy's deepest bunkers, ripping the viral blueprints directly out of our genetic code. It is an all-out dual-front assault on the disease. We are standing on the absolute threshold of medical history. There are still safety trials to complete and optimization hurdles to clear, but for the first time since the epidemic began, we aren't just talking about managing HIV, we are actively designing the tools to erase it. What do you think? Will nanotechnology and CRISPR deliver the world's first true cure? Let me know your thoughts in the comments below. Hit that like button if you learned something new, and subscribe for more deep dives into the future of human medicine. Stay curious, and I'll see you in the next video.